So we have been listening a lot about the secondary biomarkers, but we should understand what are they, because without understanding, we won't be able to understand how these secondary biomarkers work in colposcopy, or when we submit pap smear slides or histopathology material. So the secondary biomarkers are produced at different biological stages of viral activity. Simple terms that when the virus attacks or infects the cervical epithelium, then first of all, it has to meet the body's immunity, which comes in the form of P53 and retinoblastoma gene. When the immunity is weak, these two factors, that is P53 and retinoblastoma gene, is not able to control the infection, and hence the virus, HPV virus, that starts multiplication. And when this starts happening, then there's a degradation of cellular material as well as of some of the viral material. So the secondary biomarkers are the proteins that are viral or cellular gene products. These proteins are measurable in cervical samples, both cytologically and or histologically. There are certain markers which are available only histologically, and some markers they are available only cytologically, and there are some markers which are both cytologically as well as histologically available. The viral infection expresses proteins in a defined order and in different layers at different stages of the normal cell cycle. So based on this, because initially it is present, infection goes to the basal layer, then parabasal. Like this, the infection goes on increasing, and so different layers of the cervical epithelium, different types of the cellular as well as the viral degradation products are released, and based on this, these biomarkers are being used and detected, and the surrogate markers also. So biomarkers are HPV proteins, surrogate markers, that is P16 or methylation patterns. HPV infection can lead to cancer through multiple pathways, but HPV E16 and E6 and E7 gene products, they are of most importance, and they react with the P53 and retinoblastoma gene, and this is the critical point. By inactivating or activating degradation of their targets, E6 and E7 eliminate genetic surveillance and allow unchecked cell cycling, leading to accumulation of mutations and eventual invasive cancer. And this mostly happens when the immunity is decreased, and which leads to persistent HPV infection. HPV 16, E6 and E7 bind their targets with greater affinity than other types of the HPV. So you have to remember that basically Ki16 and P53, Ki16 and P16, all these molecular biomarkers are basically for the HPV 16 and 18 and not for the other types. So for other types of the HPV infection, which are high grade, high risk or oncogenic, other biomarkers are being developed. And another important problem which the scientists are facing is that because of the vaccination, the type replacement, that is the types of the high-risk HPV which is not being covered by the vaccine, they get multiplied, and they are not detected by P16 and Ki67, and that is why they are developing more and more biomarkers. And that is why, again, I emphasize there is no point in doing high-risk HPV DNA testing, because a lot of other markers and co-testing or the triage is to be used. I had already told you earlier also that the blind spot in cervical cancer screening through high-risk HPV means that we are missing HPV independent or HPV negative cancer. 5 to 11% of the cervical cancers are truly HPV negative, and 100% of the gastric type adenocarcinoma are HPV independent. So 15 to 20% overall HPV independent fraction is entirely invisible to high-risk HPV testing. Another, I have already discussed in one of my other modules, but just I want to tell you that there is a study done by Dr. Jwala, which was published in International Journal of Radiation Oncology, in which she has found that 30% of the population where she has done the study, they were HPV negative. So if so high amount of the percentage of the people are HPV negative, this means that if we go for the high risk HPV detection test only, we can miss these cancers. So the literature says that non-HPV risk factors are structurally ignored. I am not going to go in the details because I have already discussed this in another module. And the main thing is that it is all basically money involvement or the financial or the conflict of interest. Roche, Hologenic, Qiagen, they have funded HPV screening trials. Gates through Gavi and then Gates through Gavi has benefited Merck and GSK because they are responsible for the making of the vaccines. Bill Gates has got huge funding, has given huge funding to this. So there's a lot of conflict of interest, and we as doctors should rise above and understand what is basically needed to be done. Now, the classification of the different human papilloma virus testing, they are nucleic acid signal amplification test and nucleic acid signal amplification test. Just for the information, we need not remember, we should know which is the best test. Hybridization capture, invader chemistry, PCR, and the transcription mediated testing. So HC2, Servista, Cobas, OnClarity, and Aptima. I'm going to tell you a bit about which is the best high-risk HPV test, sensitivity versus specificity. So for that, Aptima is the best considered followed by the Hologic because the technology uses messenger RNA. I've already told you that the messenger RNA is a very good way, is now coming in a big way because it improves the specificity. The sensitivity is very high and the specificity is highest among the commercial tests. But only the problem is that because the messenger RNA testing is now still in the developing phase, so they can only detect HPV 16 and 18 types individually. Another is the pre-detect HPV, which again is the messenger RNA. It has got the moderate sensitivity, but the highest specificity, and it can detect all the 7 types. So the best test currently is the Aptima HPV assay, and because again it is a messenger RNA, it outperforms the DNA test in specificity, and plus these tests are able to tell that which are the on the basis of the testing, they are able to tell that if they are detected, that these might be the transforming HPV infection, which is the most important to us because we want to see whether this lesion can progress to, for example, CIN2 can progress to CIN3 or not, and this is the transforming infection. Can be done by the transforming infection only, and this is being tested by the messenger RNA. So clinical bottom line is that highest specificities of the Aptima or pre-detect test. Best genotyping is BD OnClarity, 6 individual types, or pre-detect which detects 7. We should avoid in young women HC2 and primary screening Cobas or Aptima. Both are WHO and FDA approved. And future is coming from the CRISPR-based HPV detection. So again, this thing that why to go for extensive high-risk HPV DNA testing when Pap smear, it is already proved that Pap smear is able to, when it's used as a screening tool, it is able to decrease the load of the cervical cancer, or it can help in eliminating the cervical cancer. Now coming to the HPV infection itself. So it is a small non-enveloped icosahedral DNA. Icosahedral means having 20 equivalent faces, that is triangles. It is a single double-stranded DNA molecule of about 8,000 base pairs that is bound to cellular histones, and it has got a protein capsid composed of 72 pentameric capsomeres. Capsid contains 2 structural proteins, late L1 and L2, which are both virally active. So this is the HPV 16 molecular diagram, and these are the E1 represent the early replication, E2 replication and transcription, E4 viral release, E5 immune evasion, and E6 binds the P53. We have seen just now. So we need not go into very details, but somewhat about that E6 and E7 are the most active part, and these late 1 and late 2, the capsid proteins, they are being now investigated and used as a molecular biomarkers. So already I have told that E6 and E7, they reflect the initiation of the cycles and they bind P53 and retinoblastoma gene. E4 is a marker for the cell cycle completion. P16 and P53 staining patterns tend to point towards more aggressive behavior, and Ki16 is a cell proliferation marker and predicts the malignant potential. Although P63 and Ki67 don't exist in the same cell, but if they are found in the same cell, then this means that the high-grade CIN or the transforming type of the infection is present. I have already told you that lot of biomarkers are being produced, so are being tested. So one of them again in the research is the MCM, which is mini chromosome maintenance complex, and this on the basis of MCM, P16, E4, one can discriminate, rather the histopathologist or cytopathologist can discriminate between CIN2, the one which can actually proliferative type of, or which can really transform to CIN3. So these are the type of the biomarkers, and I'm telling you again and again that the science is very imperfect, it is subjective, lot of research is being done, and it is not easy for the pathologist also to comment easily. That is why in smears also they are giving ASCUS or ASCH. Again, methylation patterns are being developed, which helps in HPV methylation, adds important risk stratification on top of genotyping. I'm not going to go into the details because otherwise it will become very, very lengthy and extensive. Productive infection mostly causes CIN1 and subset of CIN2, and they spontaneous remission. So this is the biggest challenge, that if your immunity is good, not a challenge, it's a good thing, then all these infections will regress because body has got the innate capacity to fight the infection. Transforming infection represent remaining CIN2 and CIN3, for which biomarkers are needed. Transforming infections can be early and advanced transforming infections, and advanced transforming infections are characterized by high methylation levels, more than 5 years of preceding HPV infection, and lack of E4 expression. So, as I told, the HPV infection doesn't mean that the patient is going to develop cervical cancer or CIN2 or 3. It means that the patient is having latent infection, nothing can happen. So many infections are latent in the body, and then it can be productive and transforming infection. So here it is a columnar epithelium. Infection start from the basal layer initially, which represent the latent infection because the virus is not able to multiply. It is lying in a dormant condition. And when the productive infection starts, the basal layer, parabasal layer, lower 1/3 gets infected, and the virus starts multiplication, and slowly when the immunity goes on decreasing and HPV infection is able to bypass the immune surveillance, then 2/3 and slowly whole of the epithelium gets infected, which is called the transforming infection. And here the E6, E7, P16 expression increases. So this is the way histopathologists give the diagnosis on the basis of the biomarkers and cytology and histopathology. So another important thing we should remember, that on the best way to understand the relative value of cytology, histology, and these biomarkers is that we have to look at all the things. We cannot say we just look at the pathologists cannot say by looking at the markers and say ki, no, this is a transforming infection or chances of having CIN3 is very high. They have to look at the cytology, they have to look at the histology and biomarker images of normality, LSIL and HSIL. So this is a productive infection. This is the E6 and E7. You can see here equivalent to the brown, and they are present in the, when productive infection is present, it is in the basal layer and upper lower 1/3, and E4 is in the basically which represent the cell cycle completion. E4 is present and detectable mostly in the upper layers of the epithelium and represents the completion of the cell cycle. This protein is not part of the virus particle but is present in very high quantities during the normal cell cycle, which is a surrogate marker. So if you look at the LSIL, then within LSIL there is an early dysregulation of the cell cycle. There's an increased level of E6 and E7. You can see the higher in the higher lower 2/3 layers you can see, and P16 also starts appearing in the lower epithelial cell and is a marker of oncogenic activity, especially HPV16. So this is again problem with the biomarkers also, that they are specific to certain high risk HPV, for example, 16 and 18. So if you look at the image of HSIL, that is CIN3, you can see that the cellular activity is completely disordered. E6 and E7 is present throughout the thickness, and because the cell cycle is not getting completed, E4 is only present in the minimal amount, but P16, MCM7 and KI67 also starts appearing, and when they have started appearing, on the basis of that, the pathologists give the diagnosis of CIN3. Here I have shown in the next slide the CIN2 comparison between LSIL and HSIL and then CIN2. So in CIN2, which is also labeled as HSIL, you can see that E4 will be absent or minimal and P16 will be strongly present throughout the full thickness of the epithelium in those HSILs that are actually CIN3, but that morphologically may be considered to be CIN2. So it is a very difficult. It is not necessary that you understand, but we should, when we are reading and trying to understand the HPV infection, it is important that through the, you see through the way the pathologists look at the slides. It is for that comparison. For simplicity, I have given you here the colposcopy report, one case done by outside doctor in 2023. HPV DNA was positive for 16, and pap smear was also done, which was CIN2, that is HSIL. They did the colposcopy and they took the sweet score was not given in this patient. The acetovite lesion was seen, grading was also not done. Iodine negative area was present, so they took the biopsy at 3 and 6 o'clock position, and no abnormal vessel was seen. So, but they did the leap at the same sitting, took the biopsy at the marked area, and did the cauterization with the ball. So, I wanted to tell you that what was the histopathology. Chronic cervicitis with squamous metaplasia and focal CIN2 and 3. So this was the report given, and then pathologist said IHC P16 to confirm, which came out to be positive, and because they have already done the leap, so procedure was complete and patient was treated. So management decisions depend upon your case characteristics and the risk assessment, then colposcopic triage tools, histology, biomarkers, and colposcopic finding, TZ type, suspected diagnosis. On these, you have to take the decision. Then in colposcopy, a lot of advancements are coming. AI based and machine learning is there, but I don't prefer because it will kill our skills in doing the colposcopy. And what is basically AI? AI is that they are putting 5,000 or 7,000 images, and on the basis of that, images, AI is able to give the diagnosis. But every patient is unique. They have got a different type of infection. They have got different type of the colposcopic images. So 100% reliability on AI-based colposcopy should not be kept, and we should always go on involving our skills in colposcopy. Novel imaging techniques are being developed, for example, narrowband imaging, and researchers are exploring the use of optical coherence tomography to provide real-time, high-resolution images of the cervix and potentially replace the need of biopsy in some cases. HPV vaccination, we all know how from how it is to be given, what is the history from 3 to 2 to 1. You know, that initially 3 doses were said, then 2 doses, and now India has already rolled out HPV vaccination in adolescent girl, and 1 dose is now it is being said. Although Foxy wanted 2 doses, it was pushed by Dr. Sharda Jain to 1 dose only. Then 90, 70, 90, we all of us were very well know that 90% should be vaccinated, 70% should be screened. Rather, I would say 100% should be screened and no vaccination, and 100% treatment should be offered. These slides which I'm going to show, they are very, very dear to me because no one knows the limitations of the vaccine. Vaccines are given to healthy people for the prevention of the diseases which they might never get in their lifetime. So vaccines need to be held to the highest safety standard possible. That is what the HPV vaccine do not replace currently available methods to screen or treat cervical cancer. Screening is must, and effectiveness of vaccines in preventing any cancer death will not be known for several decades. That is the biggest flaw. First flaw is that the side effects are not monitored, and then second, all the studies, they have not studied the cervical cancer as an endpoint. So the effect of the vaccine in decreasing cervical cancer will not be known for years. Rather, everyone will forget that this particular girl has received the HPV vaccination once she develops certain problems. Because suppose a girl of 14 is vaccinated, and after 15 or 17 years she develops primary infertility or premature ovarian failure, no one is going to link it to the HPV vaccine. Moreover, death rate from the cervical cancer in 19 to 20 old girls is 0, and giving the side effects in that age is not required or not done at all. So these are the 9 factors which I have enumerated here, which have been not recognized in the HPV vaccination trials. One is CIN regression, HPV negative cancer, cervical cancer, adenocarcinoma, non-vaccine HPV types. Means vaccine is covering only, nanovalent is covering only 7, and quadrivalent is covering only 2. Remaining 12 are still present. Then there's already a lot of studies coming which are saying that there's a type replacement. Means if we give the quadrivalent or nanovalent, the other high risk type, they will start multiplying, and patient who has been vaccinated might have higher chances of developing the cervical cancer because of other replacement by the other types, that is, which are not being covered by the vaccine. So waning immunity is not being studied in the vaccine. Not being studied means endpoint, that is the cervical cancer development is not studied, and it has been almost 100% proved that the immunity is waning after 12 to 15 years, 50% of the population. So that is another big problem. And there are risk factors we have already discussed. They are not being, even when they are present, vaccine is of no use. And then the safety profile, because of the aluminium adjuvant, and Gardasil 4 and 9 has got a novel adjuvant, that is AAHS, and there's lot of conflict of interest which I have shown you earlier also. So what they are saying is that vaccination will prevent 70 to 80% of the cervical cancer, but in if we do the arithmetic on the points which I've discussed just now, it might not even prevent 15 to 30% of the cervical cancers. There's a lot of conflict of interest. All the studies, trials, they have been funded by the Merck and GlaxoSmith, and they have not studied the endpoints properly. These are the 70 to 80 articles or published in the different journals regarding the side effects or the toxicity of the vaccine. India has got a very poor adverse event reporting system, and it is being suppressed because of the government initiative also. We should also remember all these things. There are few retracted papers, which is of very, very concern, because it has been found very clearly that whosoever speaks against a vaccine, they are either removed from the job or suspended, and the papers are very quickly retracted because of the vaccine media plus the pharmaceutical company. There are a lot of cases going on. And we know that Japan had declined immunization, HPV immunization. It has stopped, but it was resumed under great pressure. And in 2024, their editorial board of gynae and gynae oncology has published that if we give the HPV vaccine between 25 to 44 years of the women, annual total and cancer deaths might increase as follow. Compared with the annual cervical cancer deaths, that is 516 in 2022 in Japan, there will be 24 times, at least 10 times more from all causes, and 12 times, at least 4 times more than from any cancer, because healthy vaccine bias, they have put a lot of focus on the healthy vaccine bias, which is not being noted, or which is not being held accountable in all the trials. Another fact, that the FDA safe limit of aluminium is 25 microgram intravenous, but a study has been done in which it has been found by the age of 13 months, average Irish infant received 5,185 microgram of the aluminium, and aluminium is neurotoxin, carcinogen. It causes Alzheimer, Parkinsonism, brain degeneration, and autism spectrum disorder, which are all increasing. So as a doctor, we are liable to understand that what we are giving to our children and coming. Lot of Indian-made Cervarix has problems. No carcinogenicity data, no mutagenicity data has been done, no fertility studies have been done, no studies on the pregnancy or lactating mothers. Even the Cervarix, that is the Gardasil 4 and 9, they don't have the data for the carcinogenicity, they don't have the data for the mutagenicity. So all these are the problems, and as a doctor, we should be very well aware of them. So another important is, there are certain questions raised by scientists, that what about the serum antibodies produced by natural infection? Do they provide protection after reinfection with same type of HPV? No, not much answer is regarding that, but if it is present, it can protect from the HPV infection. And does a DNA negative state represent complete virological clearance or immunological control of infection below detection limits of the current HPV assays, which we have seen? Might be the RNA type of the testing be able to detect the infection which is present at a very low level. This is from the USA, which is a National Vaccine Information Center developed by some people to tell what has happened regarding the side effects. So as of May 1, 2026, there are 1,041 claims have been filed to Federal Vaccine Injury Compensation Program for 24 deaths and 1,017 injuries that occurred after HPV vaccination. Of that, 217 claimants have received the compensation. So the key message is, world over, those scientists, those who are not in favor of vaccination, and it has been produced repeatedly that we have good evidence that cervical screening significantly reduces the risk of cervical cancer in women, regardless of whether they have been vaccinated. The number of new cancer and deaths has decreased markedly such that cervical cancer now accounts only for 1% of the cancer deaths in women in UK, and that is why again the vaccination is not needed. These are the books on the side effects and how the girls have suffered after the vaccination. One should at least read the HPV vaccine on trial, and this I have read completely. And so many cases are there mentioned and how they are fighting legally to get the compensation. So the take-home message is oncogenic HPV tests determine the presence or absence of the virus particle, and the HPV infection can be latent, transient, productive, or transforming, and doesn't mean that the women will, once infected, will develop the precancerous lesion or the cervical cancer. And we have got a long list of the secondary biomarkers which are produced at a different biological stages of the activity. More and more are coming and get flooded in the market. The overexpression of P16 is a marker of oncogenic activity for the HPV type 16. Ki67 is a cell proliferation marker and predicts malignant potential. It is a better marker than P16. It is only for the CIN2 lesion where biomarkers are required. Vaccination cannot eliminate cervical cancer. That is my concern, and I hope I am able to make all of you understand that vaccine is of no use. Till you keep the data and you follow the girls at least for 40 years. That is my request to all of